The evolution of the eye has been a subject of significant study, as a distinctive example of an analogous organ present in a wide variety of taxa. Complex, image-forming eyes evolved independently some 50 to 100 times. Complex eyes appear to have first evolved within a few million years, in the rapid burst of evolution known as the Cambrian Explosion. There is no evidence of eyes before the Cambrian, but a wide range of diversity is evident in the Middle Cambrian Burgess Shale, and the slightly older Emu Bay Shale. Eyes show a wide range of adaptations to meet the requirements of the organisms which bear them. Eyes vary in their visual acuity, the range of wavelengths they can detect, their sensitivity and low light levels, their ability to detect motion or resolve objects, and whether they can discriminate colors. History of research. In 1802, philosopher William Paley called it a miracle of design. Charles Darwin himself wrote in his Origin of Species that the evolution of the eye by natural selection at first glance seemed absurd in the highest possible degree. However, he went on to explain that despite the difficulty in imagining it, this was perfectly feasible, if numerous gradations from a simple and imperfect eye to one complex and perfect can be shown to exist, each grade being useful to its possessor, as is certainly the case, if further, the eye ever varies and the variations be inherited, as is likewise certainly the case, and if such variations should be useful to any animal under changing conditions of life, then the difficulty of believing that a perfect and complex eye could be formed by natural selection, though insuperable by our imagination, should not be considered as subversive of the theory. He suggested a gradation from an optic nerve merely coated with pigment, and without any other mechanism, to a moderately high stage of perfection, giving examples of extant intermediate grades of evolution. Darwin's suggestions were soon shown to be correct, and current research is investigating the genetic mechanisms responsible for eye development and evolution. Modern researchers have been putting forth work on the topic. D. Nielsen has independently put forth 4 theorized general stages in the evolution of a vertebrate eye from a patch of photoreceptors. Nielsen and S. Pelger published a classical paper theorizing how many generations are needed to evolve a complex eye in vertebrates. Another researcher, G.C. Young, has used fossil evidence to infer evolutionary conclusions, based on the structure of eye orbits and openings in fossilized skulls for blood vessels and nerves to go through. All this evidence adds to the growing amount of evidence that supports Darwin's theory. Rate of evolution. The first fossils of eyes that have been found to date are from the Lower Cambrian period. This period saw a burst of apparently rapid evolution, dubbed the Cambrian Explosion. One of the many hypotheses for causes of this diversification, the light switch theory of Andrew Parker, holds that the evolution of eyes initiated an arms race that led to a rapid spate of evolution. Earlier than this, organisms may have had use for a light sensitivity, but not for fast locomotion and navigation by vision. It is difficult to estimate the rate of eye evolution because the fossil record, particularly of the early Cambrian, is poor. The evolution of a circular patch of photoreceptor cells into a fully functional vertebrate eye has been approximated based on rates of mutation, relative advantage to the organism, and natural selection, based on pessimistic calculations that consistently overestimate the time required for each stage and a generation time of 1 year, which is common in small animals. It has been proposed that it would take less than 364,000 years for the vertebrate eye to evolve from a patch of photoreceptors. One origin or many. Whether one considers the eye to have evolved once or multiple times depends somewhat on the definition of an eye. Much of the genetic machinery employed in eye development is common to all eyed organisms, which may suggest that their ancestor utilized some form of light-sensitive machinery, even if it lacked a dedicated optical organ. However, even photoreceptor cells may have evolved more than once from molecularly similar chemoreceptors, and photosensitive cells probably existed long before the Cambrian explosion. Higher-level similarities, such as the use of the protein crystalline in the independently derived cephalopod and vertebrate lenses, reflect the co-option of a protein from a more fundamental role to a new function within the eye. Share traits common to all light-sensitive organs include the family of photoreceptive proteins called opsins. All 7 subfamilies of opsin were already present in the last common ancestor of animals. In addition, the genetic toolkit for positioning eyes is common to all animals. The PAX6 gene controls where the eye develops in organisms ranging from octopuses to mice to fruit flies. These high-level genes are, by implication, much older than many of the structures that they are today seen to control. They must originally have served a different purpose, before being co-opted for a new role in eye development. Sensory organs probably evolved before the brain did. There is no need for an information processing organ before there is information to process.